Morphological features, these are categorized into 4 types, that is yeast, yeast-like, mold, and finally dimorphic fungi. Okay. So based on the morphological features, these are categorized into 4 types. Yeast. So these are round to oval cells that reproduce by an asexual process called budding, in which cells form protuberances which enlarge and eventually separate from the parent cell. What happens in yeast? The daughter cells separate from the parent cells. Examples include Cryptococcus neoformans. This is the most pathogenic yeast, and the Saccharomyces cerevisiae, which is a non-pathogenic yeast. These are the examples of yeast. And yeast-like, which is this yeast-like in some yeast, example Candida, the bud remains attached to the mother cell. Okay, in yeast, the bud separates, the daughter cell separates from the parent cells, whereas in yeast-like, the bud remains attached to the mother cell that elongates and undergoes repeated budding to form chains of elongated cells called pseudohyphae. Okay, the bud remains attached to the mother cells and produces chains of elongated cells called pseudohyphae. So this is the main difference between yeast and yeast-like. So they can be differentiated from true hyphae. So these yeast-like group has pseudohyphae. That pseudohyphae can be differentiated from the true hyphae because they have constrictions at the septa. As they have constrictions at the septa, I will show you in the picture. Here you can see. So this is a image of yeast. Okay, budding yeast. Okay, this is a mother cell, this is a daughter cell. It remains attached to the mother cell and finally gets separated from the mother cell. Okay, in pseudohyphae, these are elongated chains of budding cells. So it produces pseudohyphae. It has constrictions at the septa. Whereas in true hyphae, there are no constrictions at the septa. Okay, this is the major difference, main difference between hyphae and the pseudohyphae. That is true hyphae and this is pseudohyphae. Molds grow as long branching filaments of approximately 2 to 10 micron width called hyphae, and hyphae can be either septate or non-septate. Septate, that is, they form transverse walls. Okay, whereas in non-septate hyphae there are no transverse walls and they are multinucleated, that is inocyte. Hyphae grow continuously and form a branching tangled mass of growth. Okay, it grows continuously and form a tangled mass of growth, that is called mycelium. So these are the basic terms. So this is a picture showing septate and aseptate hyphae. Here you can see the septate, okay, the transverse walls. Whereas in sinusitic hyphae, there are no, they don't form transverse walls. Okay, they are multinucleated. These are multinucleated sinusitic hyphae, which is non-septate hyphae. This is a septate hyphae. And this is a tangled mass of hyphae. Okay, this is called mycelium. And based on the growth pattern in the culture medium, the mycelia may be categorized into 2 types. One is aerial mycelium and other one is vegetative mycelium. Aerial mycelium is nothing but it projects above the surface of the culture medium. Whereas in vegetative mycelium, what happens? Here, the part of the mycelium that grows on the surface of the culture medium. Okay, here this in aerial mycelium, it is a part of the mycelium which projects above the surface of the culture medium. Here, this grows on the surface of the culture medium. So this is a difference between aerial mycelium and vegetative mycelium. And molds reproduce by formation of different types of sexual and asexual spores. Examples of molds includes Aspergillus, Penicillium, Rhizopus, Mucor and all. Okay, these are filamentous fungi.